Dear viewers, at the outset, I would like to thank you all for the overwhelming response that I have received from you all. I request you to subscribe to my channel. In this presentation entitled The Tragedy or Comedy of Errors, I would like to take you through a very important aspect of medical laboratory workflow, namely the pre-examination process. A process which by its very nature is prone to errors which can have a huge impact on patient care. This poem looks at some typical examples of such errors. The title is Tragedy or Comedy of Errors. However, maybe there is nothing comical about it. These errors are of a serious nature. This poem has been published in the year 2011 in the journal Clinical Chemistry. I would like to start with a proverb. Well begun is half done. This is applicable to medical laboratories as well. So, let us commence at the very beginning before we actually start performing our tests. Let's look critically at the pitfalls and the potholes. Let's subject our pre-examination processes to an inquest. We'll look at some common scenarios that we encounter day in and day out. The risk or the possibility of harm to the patient from an erroneous result is what it is all about. Errors at patient registration to identify as not being used. By accepting request forms which lack clarity, a cascading effect might be produced. When a fasting glucose is requested and the patient has had a small bite or a postprandial sample collected after a breakfast that is too light. Wrong labeling of samples, inappropriate collection time, drawing blood from a vascular access device when an IV fluid is down the line. When the ratio of anticoagulant citrate to sample is not 1:9 as required for the coagulation tests, the results of PT and APTT tests become unreliable. On fresh samples drawn, you would have to retest. Samples for urine culture and sensitivity collected in a non-sterile collection device, not following the order of draw as required. The results of several tests you might jeopardize. The erythrocytes slap up glucose. Blood glucose levels will slowly fall unless an inhibitor such as fluoride is added so that the process of glycolysis it can stall. Use of wrong collection tubes like EDTA ones for electrolytes with blood collected into plain tubes, you could never get HbA1c values right. Sending blood for arterial gases collected at the venous site, inadequate mixing with heparin, air bubbles visible to plain sight. Samples forced through the needle, causing it to be hemolyzed. Samples of insufficient quantity or after a long gap analyzed. Samples meant for labile measurements such as intact PTH, ammonia or lactate not being sent on ice packs and reaching the lab too late. Microclots in the specimen block probes and impede smooth flow, might even lead to erroneous results as sample volumes aspirated would be low. Vigorous shaking of samples, extended spinning times, malfunctioning centrifuges which sometimes stutter and whine. Transferring samples after centrifugation from the primary tube to another could lead to a mix-up of samples and spoil the results altogether. Prolonged exposure to bright light of icteric samples of neonates causes ex vivo photodegradation. Serum bilirubin values deteriorate. This report is sheer nonsense, is a comment that's sometimes heard when the doctor looks at the report and thinks that the results are absurd. In the lighter vein to our esteemed end users, you will have to read or tread with caution before making such a remark. But for these lab reports, you will be groping in the dark. Elementary, my dear Watson. Sherlock Holmes would surely say, "You missed a crucial point, dear Doc, and that's the cause for your dismay. Are you sure that the sample was collected from the patient whose name you wrote down? Was the sample languishing in the ward for hours? Inquire before you fret and frown." The pre-examination phase is critical. It's patient safety which is at stake. Let's keep this a top priority and ensure that good laboratory practices we shall not forsake. So now we reach the take-home points. What do we learn from this presentation? The first one is the laboratory testing cycle can be conceptually divided into three phases: the pre-examination phase, the examination phase, and the post-examination phase. The pre-examination phase starts with the request for the tests made by the clinician and ends just prior to the sample being taken up for the examination or for the analysis. Most of the errors that occur in clinical laboratories have been found to be associated with the pre-examination phase. The major stages of the pre-examination phase in chronological order are as follows. First, we have the test request form preparation. Then we have the patient preparation. Then the registration of the patient. Then we have sample collection. The sample is then transported to the lab. The lab prepares the sample for analysis prior to loading it onto the equipment. And as we have seen, errors can occur in each of these steps. And what we should remember at all times is that these errors could have a direct impact on patient safety. So let us remember the famous words of Hippocrates. First, do no harm. Thank you for your patient listening. We look forward to seeing you again in our next session of the School of Medical Laboratory Sciences with another poem. Your feedback is solicited. Here is my email ID. Thanking you once again and hoping to see you soon.